Oh, it's a real pleasure to be here. Many thanks for the invitation. I appreciate it. I thought for this particular topic, rather than going through a block and tackling nuts and bolts catalog of where we are with rare disease and translation, I'd try to stay to the theme of the meeting and talk. I'm a geneticist and have been chasing genes and the translation of those genes into medicines my entire career. Stay with the theme of the individualized medicine and rare diseases, and actually the interplay with common diseases. So that's where I want to take this talk today. And if we want specifics about the status of the field in rare diseases, we can take those up on question time. So I don't know where people's perspective here are in general, but the genetics community at the moment, the academic genetics community, is pretty excited and about the findings that are coming out and doing a lot of victory laps in terms of all the great successes, justifiably, that have come out. But I think it's good to start with a little bit of a dose of reality on the translational side. And I tend to start with this. That's a picture of the hype cycle for those of you that don't know it. I love the hype cycle. I think all of our technologies go through it. At the top are some of the seminal discoveries that we have encountered in the field from rare to common diseases. I bring the top part up there to remind you that the cystic fibrosis treatments that were discussed earlier in this session and are so exciting and wonderful were 30 years in the making. And right now, the genes that were found just behind that, Duchenne, Huntington, even around that PKU, we're just in the clinic now for those, for treatments. It takes a really, really long time to get through, or it has taken those. So most of the things that you'll see in the clinic for rare and for common diseases today don't have the benefit of the recent genetics discoveries because they take so long to discover. So all the GWAS data, they're not even we're not even in there yet. Those were discovered 10 years ago. So they're just now finding their way into practical reality. And the bottom hype cycle slide is worth knowing because those of you that don't work in the translational setting, in the the discovery of the early monogenics into the Human Genome Project around 2001, 2002, 2003, industry invested really, really substantially in the industrialization of drug discovery, thinking if only we could just find the genes, then out will come the targets and out will come the medicines. And when that didn't turn out to be an accurate prediction, a lot of industry left, right? And it really was the hype cycle, and the funding dried up, and it was not the type of thing you wanted to talk about in polite conversation. Everyone ran away from genetics. Just about the time everyone's gone, it starts to reveal that it is actually productive and useful, and that is the hype cycle. And that's what's happening, I think, with the field right now with genetics. I want to start with that so we have a baseline going into translation. Now, if you think of rare disease gene discovery, it's phenomenal now with respect to, mostly on the basis of next-gen sequencing. So again, turn the clock back 30 years, and it took how many years and how many teams to find CF and Huntington's and so on, and the big family collections that were, remember the Venezuela family collection that you could put the pedigree across the entire wall. So now this paper's a bit out of date now, and I don't know if there's an updated one, but last time I checked, there were 3 genes discovered for rare diseases per week. So that is phenomenal, right? So the data are coming. Rare diseases are really, really leading the way. And actually the regulators, you're not meant to really digest this other than to see that the shape of these curves are going up. The regulators FDA that is, and the Europeans, are becoming more flexible and realistic about rare diseases and not forcing them to fit into common disease frameworks of very large placebo-controlled trials that are often completely impossible for rare disease settings when you need to do that. So things are lining up for rare diseases in a super positive way. Now, I said I wanted to talk about the interface between common disease too. Common diseases are really interesting, so I won't need to go through GWAS. We've heard about those here in various guises from all of us in previous talks in this session. The first GWAS findings were in 2006, 2007, so only 12, 13 years ago. And for those of us that were in the gene finding field before that, we were sitting there at meetings like the American Society of Human Genetics or satellite meetings, having really vitriolic debates on whether or not there were 10 genes known for common disease, or 8, or 12, or somebody really bullish might say 15. And it was ApoE and BRCA1/2, and these were the ones. And I was a PI on the first big GWAS study, and we submitted that grant to the Wellcome Trust in the UK. I was in Oxford in the UK, and we submitted that as a pilot study to see if there exist genes for common complex diseases. And that's why that particular study had a whole bunch of different diseases, because we didn't know which disease to start with, and we thought if we could get enough of them, then if there exist genes, one of them will have something in there, and large sample sizes, and so on. And so now you look at these, I don't know, someone could correct me, but I bet there are 6,000, 7,000 loci in the GWAS catalog. In 10 years' time, in 12 years' time, absolutely phenomenal increase in information in genetics, both rare and common. The problem, I guess before I go into this, the problem with this, with the common disease setting, is in a translational sense, there's now, you've gone from a paucity of good drug targets that have strong human genetic evidence that they are useful to pursue. Most of the drug targets that people pursue are actually based on distal animal models or expression data. That's actually what's mostly going on outside of cancer in pharma. mRNA expression data and some animal models. Pick your rotarod, and that'll carry a lot of the translational settings. Then you put this into play, and you have 250 potential loci for type 2 diabetes, and it's paralytic. No one knows where to start. We went from having virtually nothing very good to so many, we can't unpack it, and we don't really know how to get there. That's where the state of the field is right now. That's the translational context in which we find all these wonderful discoveries, and we're kind of stuck. Certain pathways get highlighted and so on, but it's really, really difficult with all that information. So that's the sobering part. Now I'll turn into try to be a little bit more upbeat and positive. I actually think this is the perfect time for translation, because we don't have really any excuses anymore. I think we've got all the pieces we need. We just have to figure out how to use them and put them together. We have all these potential drug targets, both rare and common. We've got a ton of genetics to deal with. Arguably, more and more and more on the GWAS front is great, but how much more do we need? We can't even digest the information that we've got today. What no one has really talked about here, because it's not a very, or I haven't heard it talked about, it's translational, but there used to be a concept called the druggable genome, which was things we could actually go pursue. The whole concept's kind of gone anymore, because we have gene therapy that we can change. We have oligonucleotide therapies. We can get to just about any part of the genome that we want to. So all these findings are actually pursuable if we can figure out which ones we want to pursue. And incidentally, each one of those technologies, I would argue, had their own hype cycle. They're 20 years in the making. A lot of investment, a lot of excitement, crash down, and just about the time everybody leaves. We can see it with oligonucleotides. We can see gene therapy is exactly that. It's 20 years old. But they're there now. And then the piece that you might not expect this talk to highlight, but I think is really important, it's been coming up here, and I'll come back to this why. The EHRs are a key link to advancing the rare and the common disease genetics to translation. And I don't know, I think you might have expected better clinical trials or new discoveries or something. Actually, I think those databases are going to be the ones that make this possible. And I'll come back to why I think that is. So again, the pieces are all there. We've got to just figure out how to make this work. Now, why does that matter from a genetics perspective? A few years ago, I was at a big pharmaceutical company. I'm at a rare disease company now. I was at Glaxo, and we put this paper out in Nature Genetics, and it actually got quite a lot of attention amongst the industry people. And the study was really simple. It was a bioinformatics exercise to take all past drug trials that failed or succeeded, and just look at those and put them into two categories. Those for which the drug target had some evidence, genetic evidence, for being associated with disease. Right? So you had some genetic, it's all retrospective, but it's as good as you could do. So those that are genetically validated, if you will, versus those that aren't. And you ask, what's better probability of success? And what this exercise showed was that for common diseases, which are here, It doubles your chance of success in a drug trial. Now, the oft-cited figures for drug discovery in common diseases is like $2 to $4 billion. So doubling your chance of success is quite an astonishing thing. That's really big. And in fact, even to this day, genetics, evidence for some form of genetics for the drug target is the biggest predictor of drug success above anything else except whether that target has already been drugged. It's a me too. It's the next biggest thing, and that's for common diseases too. But look at the situation for rare diseases. Could we move into that situation? It's probably unsurprising that Mendelians and monogenics are more likely, if we can get to the core etiology of the disease, more likely to be successful, and they are. In fact, 10 times more successful. So the rare disease setting is a much better place, a much better ground to plow than is the common disease setting even, from a translational setting, if you could. So that's the background. Now I just want to give you some examples and head into this EHR situation. So what are we looking for? Again, set the context. In translation, we've got all this information and we're kind of stuck because we often don't know where to start. We have hypotheses and they're really expensive studies, and in translation, in drug discovery, it's a really tough field because you often don't even know if your hypotheses are correct for a decade. It just takes a long time to push the information through, and you don't find out till a long while later. So what are these sort of idealized targets that we would like to work on? And PCSK9 and this so-called experiment of nature is the classic example. Just so you know, so this is your paradigmatic, if you could choose a new gene to work on for translation, it would have properties like this one. The PCSK9 has, in some individuals, a loss of function mutation associated with protection, if you will, some positive attribute. In other individuals, it has a gain of function mutation that is a negative effect. You got both ends of the spectrum in different individuals. So nature gave you the drug response. You know the features as high in the dose as you could go or as low in the dose as you could go, and it's all painted out for you. And all you have to do is make a synthetic molecule that mimics what nature's already got out there. People call it an experiment of nature. Everybody would love to pursue these type of things. Now, the problem is they're a little bit elusive, and we haven't found a whole lot of them. By the way, that one, PCSK9, has made it to market. They're Amgen and Regeneron both have marketed products. I think they've got commercial issues they're dealing with, but in terms of the drug itself, it does exactly what the genetics says, or the genetics says it should have done. So that's what we want, rare or common. We want something with attributes like this. Just to give you another example, one that's very hard to drug, but there's another target called NaV1.7 for sensitivity or hypersensitivity to pain. Same sort of thing. You've got gain of function with one attribute and a loss of function on the other. So this is where we want to go. Where are all these? Why do we only have PCSK9? There's a bone one as well, NaV1.7 for pain. When we're sitting on 6,000 GWAS loci and we've got 3 Mendelian genes coming out every week, are these just really, really rare to have these type of mutations? Or are they out there and we just don't know? Because if we could find them, again, this is where you want to spend your energy in drug discovery and your resources. So I've been in the GWAS world for a long time, and I've probably been the most, in a negative way, critic of GWAS studies of height. Not because I think height is unimportant, I think height's very interesting, but because they're the easiest studies to do. Every disease you look at, every collection of individuals measures height and weight. So the data are in abundance, and it's not a particularly intellectually inspiring exercise to just measure what someone collected along the way. But nonetheless, the studies have been undertaken with great success since the very beginning of GWAS. I'll put you, there's a ton of papers. I just grabbed a few of them here that were illustrative. The first one, 2007, right on the back of the first GWAS studies. One locus emerges for height. Next year, 20 for height. A few years later, 180. 2014, 423. Now, these are robust, real findings. Most of the GWAS data are substantial, so I'm not going to turn around and tell you these are wrong. Latest paper I saw was in bioRxiv, 712. Okay, so where do you start with that, with height? And I think there are other traits with even greater numbers, but this has got to be one with about as decorated as one can imagine, with genetics of a common phenotype. So let's ask the question, of 712 loci, how many of those look like PCSK9 and have Mendelian forms that go down, or 4? 4 out of 712, and they're all in the same pathway. FGFR3, which is the most commonly spontaneously mutated gene in the genome, or one of them, and causes achondroplasia. CNP, which modulates FGFR3. NPRB, which is the receptor for CNP. And SHOX, which upregulates FGFR3. The whole thing points to one pathway just by figuring out how to draw all those data together. Now, I can't say, and someone will push me on this, and you'll be right, that there aren't others that are not been found, because this is not proving the negative. I'm sure there are more. But to me, that's pretty astounding that you go through, you troll through 712 loci. I wouldn't know what to do with those 712 loci. And the CNP example, these guys in the GWAS type in the middle are completely nondescript. You wouldn't pick them out from anything. They're tiny, tiny, tiny effect sizes, and unless you were looking at them biologically, you'd be hard pressed to distinguish those as interesting loci. So you could argue that the CNP biology is yet another example of those that fall into the PCSK9 category. So I raise that up because, height, the most obvious GWAS study to look at, and they are in there. So the question I will leave you with in a few slides here is, how do you find these? They have to exist out there. How are we going to find those? Because you're not going to, and in fact, I should say, there are other examples, not with the great properties of gain and loss of function, but examples where there are really severe mutations in genes like lysosomal storage disorders, Gaucher disease, and less severe mutations in those same genes for more common diseases, Parkinson's for that, right? And they're pleiotropic. There are epilepsy genes for Dravet disease that have generalized epilepsy that in the same gene with less severe mutations. And how are you going to find those data? Because the rare disease collections aren't going to have the phenotypic information of the common disease. And if you go just measure Parkinson's disease, you're going to have only Parkinson's disease, and you're probably not going to have the rare diseases. How are you going to find how are we going to find any of these loci except by serendipity? I think the answer is in the EHRs, because you've got to have the broad clinical information. You don't know if a lysosomal storage gene might be associated with MS, and so you would never do the study unless you've got a population with both genetic information on the really rare diseases and clinical information on the common diseases. So I think this is where the field is going. I put these up just as an aid memoir for myself. You could put all of us up here as well. I'm on the All of Us, I declare on All of Us advisory panel and have been for a number of years. I chair the genomics working group. The UK Biobank was started in 2001, is along the same genre. It's 500,000 people in the UK, which has a nationalized healthcare system, so much easier phenotypically in terms of harmonization. I remember when this grant was, I wasn't part of it, but I was on the review, I reviewed it. When it was submitted to the MRC and to the Wellcome Trust, see if you could get funded here for this, the front line was, this study won't be interesting for 15 years. And they got the money. And the goal was it had to be prospective and just wait. Now, last summer, the data finally become available, and I think anybody in the human genetics field, postdocs' holidays were canceled, if they ever get holidays anyway. People were lined up waiting for these data to be released. I think the first paper was in bioRxiv within a week or 2 weeks. It was pre-written, just tables. So everybody dove into this, and it's been a massive success, and so will all of us, and so will all the others. Genomics England on the right-hand side is specific. It's a different program, but specifically focused on rare diseases. We're involved with that now. It's only 100,000 people, which is small, but it's got sequence data versus genotype data. We're finding misdiagnosed cases. We're finding undiagnosed cases. We're finding some really interesting biology on maternal transmission. these aren't things that need to be proven. The challenge, I believe, here is that none of them are big enough. I don't think any one of them, I don't think a million, in all of us is enough. I think we need them all, and we need to put them together. And I think that's where it's, especially for rare diseases, because any one of those, we work on diseases 1 in 50,000 births, 1 in 100,000 births, so these aren't going to really reveal enough. So I saw, and there's another meeting coming up last year about this time. There was an international cohort summit that I think Jeff Ginsburg and Francis Collins and others coordinated. But I thought this was interesting because they put together, I wasn't part of this, but minimum criteria, 100,000, you had to have 100,000 people involved, not disease specific, available biospecimens. They found 60 such cohorts. So there are 60 out there to play, to potentially bring together, and some of them I think are represented in this room. I suspect that might have been part of that. 60 cohort studies involving 25 million people, 24 countries. That's where we need to go, and that's where the translation, I think, will really come out of that. All the AI pieces, how we're going to harmonize those data and bring them, I leave it to you. Really, really important, but the genomic information is already very, very exquisite. It's the clinical and the phenotype side that we're really going to need to get our heads around. And I leave you with just the fact that the funding will find its way there. Pharma's been interested in these type of big data sets for many years. This isn't new. Amgen bought Decode now 6 years ago, seven years ago. And so the funding will follow both on translation, I'm sure, and on national podiums if only we can find a way to bring all these data sets together. So I'll conclude with just my view of the cartoon that I think the rare diseases coupled with the EHRs are going to bring us together with common diseases. And the individualized medicine is the outcome of that sort of continuum from rare to common. Thank you very much, and answer questions.